mentioning about those positions that are state
employees,
by the employees. I don't know how I can. Wait. I have something mentioning that. Okay. Voilà, c'est là. Okay, here we go. The staff scientist position in our labs, those positions, there are less and less. At the lecturer level, there are plenty of them, but CNRS position, for example, without being a PI, is now becoming very rare. And very tricky to get, and actually the level of recruitment now is just crazy. I mean, all have PI type files, so it's basically now the French system is really moving towards like permanent position are given to PIs, basically computers.
It's like a ratchet. It's becoming increasingly.
It evolved this way. I mean, that's a general trend and general move. So that's really the actual moves is there. But actually, I mean, in France there is a lot of money in research. I mean, it's rather whether it's put towards biomedical science and innovation, and what goes to basic research is, I mean, that's more about the repartition of the budget. That is really an issue rather than the budget itself. Of course, we would prefer to have like increase in budget. There was some recently, but not huge. We are still not at the level of Germany, for example, but and there is still already a lot of money put by the government in research. So it's rather of allocation of funds rather than the actual funds itself. So.
All right. Well, it's just one. And so I think it would be wise probably to wait for a few minutes just to make sure that everybody has the opportunity to join in. But well, I realize now that I have to introduce you, and I haven't really asked much about your career. I mean, everybody can read the bio in the website. But what I'd like to say for the people who are attending and will be coming in the next few minutes is that François, he's been a pioneer in studying the interactions between bacteria and the microbiota and metazoans, and in particular he started working with Drosophila. Now he's expanded, I think, into mice more recently, and I still remember when I was a young postdoc, his first PI papers describing, I mean, doing, I think the dream of a geneticist where you put two organisms together, but then you start tampering with the genetics of each of them, and then they're starting to find, signaling circuits that span two, that govern the interaction between two species. And I think that's something that it is incredibly powerful, and also puts a lot of emphasis on the role of metabolism, which I think from the point of view of genetics has been neglected. It was the land of biochemists until relatively recently. And I think now with the power of modern techniques, it is starting to become more amenable, and there's a revamped interest in it. So, and I think, well, François is probably best placed in Europe at least in Europe, to give us a talk about this, which is, I think, touches many of the research interests of the school, and particularly mine. So, without further ado, I would like to just give the floor to François and just listen to him.
Great, thanks very much. Thanks for the introduction, Joaquin, and really thank you for the invitation. I mean, we were chatting together and exchanging by emails and hoping we could have this visit on site. I mean, I was really looking after a real visit after all those months of Zoom meetings. There was a window of opportunity for a few months that collapsed thanks to Omicron. I don't know how is it in UK, but now in France it's getting a bit better. So we are reconvening some meetings and stuff like that, but it's really the early days. So I mean, we have to do that by Zoom today. I'm sorry about that, but at least we have some opportunity to discuss after the seminar with few of you. So yeah, so thanks for the intro, and yes, I've been working on this model of host-microbe interaction, and I will illustrate. I mean, I decided to do a bit of an overview in my seminar and sharing some latest results at the end of the seminar and publish results on our mouse work, but also few fly work that we recently published. But I decided to start it with somehow the kind of the whole story of the last few years, starting from the initial publication you mentioned, Jack. So yeah, I mean, we work on the microbial influence on host biology and basically host physiology. So wait, I need to start that, and that should work. Yes, so we basically have a host-centered view of host microbe interaction, and we are very much interested in understanding how the host physiology, the host biology is shaped by the environment. And when I mean environment, I mean the nutritional environment and the connection between nutrition and host physiology and host biology is very well established. I mean, we don't know everything, but I mean, there is strong connections there. And what we are interested in my lab is to study actually how the microbes, the environmental microbes, but also the internal, the intestinal microbes, the microbiota, shape this interaction between nutrition and animal physiology. And as Joachim said, I mean, we are focusing on partly on metabolism, but I mean, our prime readout is to study juvenile growth. So the juvenile phase of the life cycle when the animal reach maturity, sexual maturity and maturation. So we are applying this conceptual approach in a specific environmental context, nutritional environmental context, which is known to strongly and severely impact the growth of the animal, which is chronic undernutrition. So chronic undernutrition is defined as a reduced dietary protein intake over a long period of time. So we are not speaking here about those acute severe famine that are triggering wasting and acute undernutrition. We speak about this reduced intake over a long period so that kids or animals that receive this condition actually have reduced linear growth and they carry immune and neurocognitive deficits on the long run, on the long term, even if the nutritional environment becomes better. And that is coined under the term stunting. So the animals and kids, they are short in stature in this context, but they develop somehow fine and they just have some deficits over the time. So we apply this host microbe interaction study in this nutritional context, and it's also because it's still very severe conditions for human. I mean, you have to realize that we all speak about diabetes, obesity in the world, but I mean in the latest report. So I forgot to update the slide, but we had the 2021 Global Nutrition Report, and it's still about 150 million kids which are stunted in the world. As you can see on this map, we have most of the prevalence is really high in Africa and Southeastern Asia. So, you know, it's really a condition that is really still very pressing. So we study, so we have a very basic approach to the problem. So we are not studying clinical conditions and we are not studying humans in this context, but we are using models and animal models to really study those interactions like nutrition on the host physiology and microbes impact on the link between nutrition and the host. And for that, we use gnotobiotic model. So we started with gnotobiotic model, and you'll see at the end of my talk, we are now working on microbial intervention conclusions on conventional animal models. So what I call gnotobiotic models are animal models for which the microbiota, so the microbial environment, is actually controlled and experimentally controlled. So for example, we have like gentry flies that we can reassociate with a defined community or defined members of the microbiota. And we have been also translating some of the discovery we made in the fly using gnotobiotic mice, so germ-free mice, which are reassociated with single or communities of known and isolated microbes. So this gives you the ability to actually control very well the microbial parameter in your experimental system and see how the microbes shape the biology of the animal under study. So I'll start with the fly part, and you'll see we've translating some of the results obtained in flies in mice. So, I mean, of course, as you all know and expect and acknowledge, I guess, flies is really an excellent model system to run genetic studies. It has gigantic experimental tractability. I mean, that's obviously not the only model for having this tractability, but it's really, the genetics is really well advanced. So beyond that, I mean, it's also a model for our specific question that is very useful because it's comparatively very easy to generate gnotobiotic animals as compared to mammalian models, vertebrate models, okay? So deriving germ-free animals is pretty easy, germ-free flies, and keep germ-free flies in the lab and reassociate flies with members of the microbial community associated to those flies. It's fairly easy in the lab and cost effective. So the microbes associated to fly colonies in the lab are well characterized. They are mostly aerotolerant. We can isolate them. We can manipulate some of those species. And community is really simple. It's really a handful of strains in the lab. In the wild, the communities are a bit, sorry, the communities are a bit more complex. But most of the dominant phyla are illustrated here with minor component of Enterococcaceae and Enterobacteriaceae, and major component of the fly microbiota are Acetobacteriaceae and Lactobacillus. Those guys and the composition of this microbiota is really influenced by the nutritional environment of the flies, and also in the lab, very much influenced by the type of preservant that you put in your fly culture. So for the fly people in the field here in the audience here, this is really, if you put nipagine or propionic acid, both or only one of them, this influences very much which type of microbes, bacteria you'll get in your fly tubes. So yeah, I mean, what is interesting also is that the life cycle of the fly is very short. We can strictly control nutrition microbiota by associating germ-free flies with different members of those communities, but also we can derive some very controlled nutritional context. I'll expand on that later using oleidic diets, so chemically defined diets. So as fly people know, when you breed flies in the lab, when you provide them with optimal nutrition, you reach the entire life cycle in about 10 days. And what was very much explained over like the last 40 years is that if you have all tubes, or actually if you decrease the nutritional value of your fly food by just diluting the fly food or diluting the content of the yeast in your fly food, the yeast extract, which is the core source of vitamins and proteins in the fly food, you see that the time that the larvae and the egg takes to reach the metamorphosis is really increased by really high factors when you dilute the yeast concentration. And up to a point where you actually have adults emerging, still emerging in this context, but the size of the animal is reduced by 60% towards the size of a well-fed animal. And that is clearly a prototypical phenotype of chronic undernutrition, a stunting phenotype, where you basically reduce the protein intake or the nutritional intake of the animal. Then you lengthen the developmental time, and you actually have adults that develop. They are proportional in terms of wings, legs, and different organs, but they are really delayed and they are really small. So we wanted to investigate in this particular context, this nutritional challenge, whether commensal microbes, so microbes associated with flies, living together with flies, have an impact. And this is the paper, I guess, Joaquin was mentioning in his introduction, where we showed 10 years ago, a bit more than 10 years ago, that the microbes which are associated to the flies are actually helping the flies to develop in such a nutritionally scarce condition. So if you take a germ-free or a conventional fly stocks with its microbe, or a germ-free microbe, actually when they're well fed, you still reach adulthood, they still reach adulthood in about 10 days. So there is no major impact of the microbiota when the animals are well fed. There is a bit of a difference, but it's not obvious. In terms of growth, in terms of aging, this is different. So when you enter with germ-free animal, you see that you lose 5 additional days. So it takes 5 days, so an increase by 50% of the developmental time. And then when you reassociate germ-free animals with specific isolates of those microbes that are associated, and especially one of the species we characterize and we use as a model in this system, one of the Lactobacilli we isolate, Lactobacillus plantarum, you regain 3 days of development. So you have a phenotypical window between associated versus non-associated animals of 3 days of emergence for a total of 12 days. This is really big phenotype in this context, which is only due to the presence of the bacteria. So that was super exciting as a result. So you see germ-free larvae that are 7 days old. They are really tiny. They develop very slowly. They grow very slowly. And mono-associated animals are really larger, really bigger. And we follow the size of the larvae over time. So the germ-free until pupillation, you see that they take a lot of time. The growth rate is reduced. But the growth period is actually also increased. And when you have association with this strain, you see that both the growth period is reduced, but also the growth rate of the animal is increased. So there is something here that the microbe is doing to help the larvae develop despite this poor nutrition. And that was really that was the beginning of the adventure of trying to understand what are the microbial cues and the host mechanisms that are involved into this phenotype. So here, over the last few years, we approach those questions using genetics, mostly starting on the fly side, and we characterize from signaling pathways expressed in enterocytes, which are really important for sensing commensal microbes, like sensing the peptidoglycan, so which is a component of the cell wall of the bacteria. And we could show through genetics that peptidoglycan sensing actually engage immune signaling, TNF-like signaling in flies, which is the IMD pathway, the immune deficiency pathway, which engage an NF-kappa B factor. And that pathway is very well known in Drosophila immunity and in animal immunity. But actually, we could show that this signaling pathway is also engaged upon recognition of commensal bacteria in the intestine, in enterocyte, and that beyond regulating immune genes and immune regulatory genes, it also controls the expression of some digestive enzymes, including peptidases, which we could show that when the bacteria is present, that signaling is activated. There is an increased peptidase activity in the enterocytes of the fly, which leads to an increased uptake of amino acids, which basically fuels the production of growth factor and growth hormone, because those endocrine cues are actually activated by sensing amino acids. And that increase in growth factor and growth hormone production and activity is actually helping the animal to increase its growth rate and shorten the maturation period. So that was established in few publications from the lab earlier on, before 2016, basically. And we exploited that phenotype by actually screening different strains, of bacteria, because we couldn't start to understand what's going on the host side, but we wanted really to know what's going on the bacterial side. So we have this assay, which is very basic, collecting germ-free animals, inoculating them on low nutrition diet, and associated them with either the reference strain we were working on with before, Lactobacillus plantarum WGL, the first strain we characterize as a growth promoter in flies. And what we did is to actually associate germ-free animal with a large collection of multiple Lactobacillus plantarum isolates, so which genome is really different. So we had also performed lots of genomics on those isolates, and we realized that the core genome of all those strains is actually really small. There is a lot of variability in the genome, in the gene content of all those strains. So we could try to associate the performance of the strain, like growth promoting strains, such as the blue and yellow ones, versus the less growth promoting strain in this context. So of course, the ability of the bacteria to grow on the fly food, to colonize the larvae, and to survive in the larval intestine is one important parameter. But it's not really what we were interested in. We were really interested in the symbiosis factor, the genes in the bacteria that are really important, not for surviving in the fly food or in the gut, but to actually trigger the growth phenotype. So we found several strains that actually colonize well, persist well in the fly food, but are less good at promoting growth, such as, for example, this LP NISO 2877 strain, which is a strain colonized very well the larvae, colonized very well the fly food. But despite that, there is an intrinsic alteration of its ability to promote growth of the fly. So we have a really strain-specific phenomena, and we wanted to understand this basis of strain specificity. So before going there, what we wanted to know is, okay, let's take the wild type strain and let's do a genetic screen, like old school loss of function screen, random screen in this context. And then when we did that a few years ago, and we generated 2,000 insertion, random transposon insertion in the genome. And we could screen for wild type strains, bacterial strains, which the insertion would alter its ability to promote entry to metamorphosis earlier than germ-free animal. And we were actually looking for mutant that would trigger delayed pupillation. So it was very easy to score. We were just counting pupils 7 days after association. In germ-free, there would be no pupae. In a control context where the wild-type bacteria would colonize, we would have most of the 40 eggs that we were inoculated that turned into pupae. And in mutant strains, we had a reduced number of pupae. So it was really easy and quantitative test. And out of those 2,000 insertions, we validated 7 insertion candidates. And among them, we had some insertion in this operand, the PBPX2 DLT operand, which is an operand that encodes genes which are important for modifying the cell wall of the bacteria. And actually decorating some of the component of the cell wall, the teichoic acid, with alanine. We knew before, thanks to the engagement of the IMD pathway and the peptidoglycan sensing by the IMD pathway in the enterocytes of the fly, that the cell wall was really important for sensing the commensals, for the host to sense the commensals. So finding genes that were actually impacting another component of the cell wall, not the peptidoglycan, but actually the teichoic acid, was somehow exciting to pursue and to dig into. So what we did is to generate clean mutation of that loci. So this is the strain where there is the insertion of the transposon, which is a loss of function. So we are looking here at larval size at day 7. Germ-free larvae are small. Larvae associated with the wild-type strain are big, like twice as big as the germ-free individuals. And you see the one associated with the transposon insertion are also altered in their growth promotion. We did some genetics by homologous recombination, and we generated clean deletion of the genes targeted by the insertion, the first gene of the operon, PBPX2, but also the entire operon. And we could see that actually the entire operon was required for the phenotype. What was very interesting in this context, and I'm making the story very short here, it's an entire paper that we published 3 years ago, we could show that the modification of the cell wall and the alanylation of the teichoic acid is actually one of the key determinants of the signals sent by the bacteria or sensed by the host from the bacteria. So peptidoglycan is sensed, but also the teichoic acid. So what we did in this context is to take germ-free larvae, associated them with either the wild type or the mutant bacteria. So you see the wild type bacteria triggers growth. The mutant bacteria has an altered ability to promote growth, and we complemented either the germ-free larvae, the wild type bacteria, or the mutant bacteria with purified cell wall. So the purified cell wall that contains the peptidoglycan and the teichoic acid from the wild type or the mutant strain. So as you can see here, and we assay larval size over time. And as you can see here, the cell wall from either the wild type or the mutant strain does not impact the growth of a germ-free individual. So the cell wall is not sufficient itself, and we'll come back to that, suggesting that other determinant of growth from the bacteria exists beyond the cell wall, and this is where I think we go into metabolism. But if you had the cell wall purified from wild type or mutant cells on the wild type bacteria, there is nothing more than the effect of the wild type bacteria. But as you can see, adding the wild type cell wall to the mutant associated larvae, so the larvae associated with the mutant that is defective in promoting growth, was able to rescue the growth promotion, while associating them with the mutant and complement with the mutant version of the cell wall, so cell wall isolated from a mutant bacteria, would not trigger growth. So that was somehow a biochemical complementation of the loss of function that was very exciting, because it tells you that you need the bacteria, so you need some other cues, which are not altered by the mutation, but you can compensate for the alteration due to the PBPX2 operant with just the cell wall. So that led us to propose this model where you have like growth promotion by the bacteria engaged by the IMD signaling pathway triggered by the digestive enzymes. And in this context, this is really a recognition of the cell wall of the bacteria through peptidoglycan recognition and also other components of the cell wall, most likely the teichoic acid and their alanylation, that is triggering optimal expression of peptidase and probably also triggering other signals. And we are now currently following that up by trying to understand how the teichoic acids are recognized by the host, how the teichoic acid signals to enterocytes, what are the signaling pathway in the host site that triggers the host response to teichoic acid. And we are now realizing that beyond peptidase, most likely other factors, other genes are activated by this secondary pathway beyond the IMD pathway, and we are actually now dissecting them. So that was the first story I wanted to illustrate that shows really that we can do genetics in the bacteria and understand more about the biology of its host. I would like just to mention another story which we published 3 years ago, where we actually showed that this symbiosis between plantarum and Drosophila is actually what we define as a facultative nutritional mutualism. So it's facultative because each bacteria, each member of the association do not need the other partner to survive in that environment. So Lactobacillus plantarum survive in fly food. Drosophila, germ-free, survive and develop in the fly food, but they are affected by this environment. And when they are together, actually, they benefit from each other. So I just illustrated that the host benefit from the bacteria, but we could show in this paper that basically the bacteria itself benefit from the host because the host is actually shedding chitin and chitin monomers and acetylglucosamine, which are super good source, super good nutrients for the bacteria. So despite a low protein environment where the bacteria has a division rate that is really low, when you have larvae in the system, and even if some of the bacterial population is lysed when it goes through the gut, the shedding of those chitin elements is actually feeding somehow the bacteria in this context and optimize this virtuous cycle of sustaining the bacteria that is actually beneficial for the growth of the larvae. So we dissected this system, and we could find this sugar coming from the host to support the bacteria. Another story, and is just I just provide the concept here. What we did is to do some experimental evolution. So bacterial experimental evolution in this system. So that was another paper we published a few years ago, where we took, you remember, this other strain that is not super growth promoting, and we actually generated a Drosophila line that was mono-associated with this strain. And we selected for 20 fly generation only the first few emerging flies from each generation. And we were thinking that we were resonating that actually the bacterial genome would evolve obviously faster than the host genome in this context. We will be able to select for variants from the bacterial genome more rapidly than from the host genome, because I mean, bacteria divide way faster than the generation time of the fly. And that's actually what happened. And we could characterize a variant of the plantarum bacterium that is actually a variant, and we don't quite understand entirely why this variant is actually accumulating and shading and releasing high quantity of N-acetylated amino acids. So that's really a metabolic mutant. It's ACK, acetate kinase A gene, which is mutated in this variant. But that mutation turns the strain, which is poorly growth promoting, into a super growth promoting strain for the larvae. And we could show that that was due to the production of N-acetyl amino acid in this context. And that was cool because it's really an additional way of getting amino acids from the diet, from the bacteria, by the host in this context. So last part on the fly system is really beyond now this genetics of the host or the bacteria. What we could do, and this is a story we published last year or the year before, no, 2020 already, and this is about nutritional manipulation, because this phenotype, it's really, as we call, a nutritional mutualism, because the bacteria helps the host to digest despite poor nutrition, and the host is actually providing nutrients to the bacteria. So now we were also thinking that there should be some nutritional, nutritional support of the bacteria to the host. So we showed that by finding that there is those amino acids that are produced by the bacteria, so there is a nutritional contribution, but we thought there would be additional contribution in this context. So for that, we turned into holistic diets. So holistic diets, those are chemically defined diets, and there was few publication, I mean, in the literature from the Micheli lab and for the Partridge and mostly Matt Piper that has design and characterize a chemically defined diet. In the 60s, there was plenty of papers in the fly field that had already defined holistic diet for flies, and actually the early days of fly nutrition was really very prolific in the 60s. So those guys somehow reinvented the holistic diet, optimized it. So we capitalized on the one developed by Matt Piper while he was in Linda's lab, and now he has his own lab in Australia. And this diet is really useful because you can really manipulate it. So what we did with few brave students and postdocs in the lab is to generate an holistic diet where each of the components were removed one by one. So the holistic diet is actually composed of 39 different ingredients, and a full holistic diet allows a development that is a bit slower than a normal fly food on jump-free animals. So you get pupae in about 10 days, while usually you get pupae in 6 to 7 days, according to the type of fly food you use. And then we remove one by one each of the component of the holistic diet. So essential amino acid, of course, animal can develop, so they are lethal in this context. Non-essential amino acid, you see that they are either wild type or a bit delayed. I will come back to this asparagine context in a minute. Vitamins, and B vitamins are really important, and I mean, flies are auxotroph to B vitamins. We could confirm that. We just reinvented what was published a long time ago. Cholesterol and choline were also very important, and then other components, including minerals, were important in this context. And what we did is to actually compare the developmental timing of jump-free animals, as I show here, with animals associated with different members of the microbial community. Just a small note about this this specific asparagine deficiency here. One could say that larvae are auxotrophic, but in fact we changed the genetic background, and we could find that this asparagine auxotrophy was really related to the yellow-white background. When we changed the background and use another fly background, we could find a delay, but no auxotrophy, no strict auxotrophy in this context. This is really for the fly people of the audience. So what we did basically is to take jump-free animal, put them either on a full holistic diet or all those 39 depleted, individually depleted diets, and we associated also larvae with either a single strain of Acetobacter pomorum, which is another dominant commensals of the flies, or the strain we were characterizing before, Lactobacillus plantarum, or it inactivated version of those conditions. And we transform that in a color code data set, so like that it's more digestible by the audience. So you can see that both strains, Acetobacter and Lactobacilli, do accelerate the development. So you have larvae reaching metamorphosis in just 8 days as compared to germ-free animal, and the heat-killed bacteria actually don't have an effect because you need the bacteria to be alive and metabolically active to support the growth of the host. So you have here the time where 50% of the population reach metamorphosis, and this is just the color code of that type of results. And then the colleagues did this great screen for a few weeks, and we could characterize actually the specific nutritional compensation that each of the bacteria was providing. So first result from that, and we'll go step by step, and that bacteria have to be alive to support growth and maturation on the full holistic diet. So that was really the first identification. This was confirming that basically we had that type of results before. So heat-killed bacteria do not complement germ-free nutritional requirement on the holistic diet. And if you compare really the heat-killed associated context, it's really similar to the germ-free context. Of course, there is a bit of exception with biotin and folate, and we believe that this is because there is contamination from the heat-killed bacteria that contain a bit of biotin and folate. So the initial input from the initial heat inactivated context seems to be sufficient. Now, what was interesting is that live bacteria complement many, if not all, of the nutritional component. Yet, it's not fully, because as you can see, you don't recover an entire survival in this context. What was very interesting is if you compare each association, clearly the nutritional compensation seems to be very specific to the bacteria. Each bacteria has its own pattern of nutritional compensation provided to the host. So you can see that Acetobacter pomorum actually has almost a full complementation pattern, but cholesterol, because bacteria cannot produce cholesterol, while Lactobacilli actually have a differential compensation. And this is very much related to the ability of each of the strain to produce those actual compounds. So that was something interesting, and making again a summary of a lot of specific case studies that are shown in this paper that for the one interested I recommend to have a look at. So axenic larvae, when they are bred on this full holistic diet, they are showing 21 auxotrophies. Okay, so 21 nutrients are really essential for its development. When you breed flies on holistic diet, larvae, plus this lactobacilli, you compensate for about 10 auxotrophies of those 21. And then what was super interesting is that this Acetobacter strain actually compensates for almost all the auxotrophy. You don't reach the full wild-type phenotype of development, but at least you compensate for many of them, and that correlates pretty well with the ability of the bacteria to produce those compounds. So that's the main compensation mechanism, is nutrient synthesis by the bacteria. But in fact, there are some interesting cases, and we are actually running a story now and a project about some functional compensation in absence of synthesis. And I'm not going into the details. This is a developing work in my lab. Hopefully it will be published in a few months' time. But for example, Lactobacillus plantarum does compensate for a reduction of valine in the diet. Yet, lactobacilli cannot produce valine. Valine is an essential amino acid also for the lactobacilli. So there is somehow some functional compensation of scarcity in essential amino acids. Of course, if you remove entirely valine from the diet, larvae would die, bacteria would die. But if you have a reduced amount of valine, somehow having Lactobacillus plantarum without providing valine its presence and some activities allow the larvae to develop faster on the low-value diet. And we've been investigating how is that functioning, and we ended up finding some interesting host microbe signaling cues in this context that we are now currently dissecting. Last part on this holistic diet and nutritional support that the bacteria is providing to the host. You've seen that we've been screening each strain independently to know the relative contribution of the different commensal members to the nutrient compensation. And then Jessica Consugra in the lab also investigating whether the two strains together would have a better effect, more than an additional effect than each strain separately. And actually, she realized there is this very strong synergistic effect between the two bacteria. And that the basis of that is that the two bacteria actually develop cross-metabolic exchanges, cross-metabolism between the two strains. And we could demonstrate in that paper that the lactate that is produced by Lactobacillus plantarum is actually utilized as a carbon source by the Acetobacter species. And that actually fuels and boosts the metabolism of the Acetobacter species, which in return produces many of the nutrients that the Lactobacilli is auxotroph for. So you see, you have this forward loop of one feeding the other and vice versa, you know, and that in this context was actually super beneficial for the host because it actually provides a full complement of nutrients to the host, allowing to compensate for many of the exotrophies of the host. So just taken together, the current model we have of fly microbes and host growth, Drosophila growth, is you have, for example, fragment of cell wall from the Lactobacilli, but also most likely cell wall fragment from the other bacteria, which are released when the bacteria is dividing in the intestine or in the diet, and that those fragments are sensed by the enterocytes and trigger those enhanced digestion mechanism I mentioned in the beginning of my talk. On top of that, you have active provision of each of the nutrients of the bacteria, active provisions of nutrients which are lacking and for which the host is auxotroph, so that helping the nutrition of the host. And beyond that, we could show that lactate addition, so the lactate produced by the Lactobacilli on Acetobacter, is actually shifting the metabolism of Acetobacter, providing more amino acids and B vitamin, but also producing many other metabolites which are enhancing host anabolism and fueling host resistance to oxidative stress. We found many phospholipids, many different lipids there that are exciting, interesting, but we don't really understand yet the connection between those metabolites and host metabolism and host stress, oxidative stress resistance. And this is the focus of a current project in the lab, where we try to follow the metabolism of the different bacteria and the host in this context by fluxomics. So this is the current working model we have with our Drosophila system. And you see that here, this is a model that is great really to dissect all the subtle mechanisms that are engaged between model bacteria and host in this context. So then what we wanted to do in parallel to this fly work is to study whether this profound effect of the Lactobacilli we characterized early on in flies has also any translation into vertebrates and mammalian models. I mean, do Lactobacilli also have the ability to support the growth of mammalian models? So for that, we generated, we set up a collaboration with some colleagues which are experts in gnotobiotic in mice. We were completely ignorant there. So Martin Schwarzer and Anna Kozakova, and now Martin, after spending some years as a postdoc in my lab, then moved back to Czech Republic. And he's the head of the institute of the lab there. And what we did is to basically reproduce in mice what we did in flies. So having germ-free individual and mono-associated ex-germ-free animal with single strains. So either the plantarum WGL strain, the one that promotes growth in flies, So having mice monoassociated with plantarum WGL or mice monoassociated with plantarum niso, which is one that has an intermediate effect on it. And what we were doing is to breeding those colonies in those isolated types, germ-free, conventional mice, so mice with a complex microbiota, and monoassociated mice with each of the strains. So we were doing that in the parents, in the adults, and we were collecting progenies and following the growth, the size, and the weight of the progenies during lactation until weaning, while they are kept with the mother on a normal breeding diet. And then we were challenging the animals at weaning by providing them a depleted diet that was expected to provide stunting, to trigger stunting. So the results were quite spectacular. So we published that a few years ago, and we could show that basically if you follow weight, but also the size, when you, let's focus on the black curve, the weight gain of a conventional animal is illustrated here. Animals don't put on weight very much when they are on the poor diet. Same for the size. You see, this is a stunting phenotype. The conventional mice don't grow as well and don't grow in terms of size as well when they are receiving this low diet after weaning. But what was very interesting is that germ-free animals actually are also suffering very much from the stunting when you are challenging them with the low diet. So you see that germ-free animals, they are almost incapable of putting on size and weight when they are suffering from chronic undernutrition as compared to mice, conventional mice that has a microbiota. So that was a first indication that beyond metabolism, at that time, metabolism in connection with microbiome and metabolism was well established. So we were expecting that weight gain would be involved, but actually the skeletal phenotype was not expected. There was no link between microbes and bone and skeleton growth. So that was the first indication. And what was very interesting to us is that we could show that the animals that were monoassociated with each of the strain behave similarly to what we could see in flies, that the animals that were associated with the WGL strain were actually behaving like conventional mice, while the one associated with the niso strain, the one that does not promote well growth in flies, was also intermediate in its ability to put on weight or actually grow in size in terms of skeleton when they are associated with this strain. So again, there was this strain-specific effect in an heterologous mammalian system when they receive each of the strains. So we decided to go further there. So it's just to wrap up that the microbiota acts as a buffer. So that was the first conclusion, that the microbiota acts as a buffer to the adverse effect of chronic undernutrition on linear growth. So, I mean, skeletal growth, linear size. We could show, but I'm not going into all the mechanistic details, but we could show that the microbiota actually sustain growth by maintaining and engaging the somatotropic axis activity, so the IGF-1 activity. We could show that IGF-1 production was restored, and if we block IGF-1 production in this context, we could wipe out the bacterial effect in this context. And we could show that selected strain of plantarum recapitulate the microbiota effect in a very conserved manner as what we've seen in Drosophila. So the next experiment we wanted to do, and this is the unpublished part just to finish the seminar. Obviously those experiments, which are heterologous by nature, okay? We take some strains isolated from flies and we put them in germ-free animals, in mice, you know. We wanted to test the potential of a microbial intervention in conventional mice. So really the logic of a probiotic intervention. So you take an animal, we design a stunting diet for conventional animals, so stunting causing diet. So this is a diet that is reduced in fat and protein. And when you have conventional animals that when they are put on this depleted diet, this time it's only 4% protein, before it was 8 for the germ-free. You have clearly a stunting phenotype that kicks in. The weight gain is altered in those conventional animals. And also the size gain of those animals after weaning is altered because of the low protein and the low fat content. Those are isocaloric content, so it's not a depletion of calorie, it's really the nutritional challenge in this context. So we did that in conventional animals. We could show that the somatotropic axis is really altered in those animals. So the circulating IGF-1 is down-regulated. The expression of markers of IGF-1 receptors expression in the muscles is affected. And the production of IGF-1 by the liver also is reduced when the conventional animals are kept on the poor diet versus the normal diet. So we, again, in this model, have an altered somatotropic axis activity. So this is the baseline setup. And what we did is to test the intervention on those animals at weaning every day until young adulthood of either a placebo preparation or the plantarum WGL preparation in this context. And what was very interesting, so we did that on large cohorts of between 20 or 30 individuals in this context, and we could show that there is a striking effect of weight gain. So a reduced weight loss, I would say, between the normal diet and the poor diet when they are treated with the bacteria. So they actually put on weight a bit better when they are submitted to the poor diet. And also the size gain was really significantly impacted by the addition of the strain in a conventional mice, so a mice that has a microbiota already. And we could quantify that in terms of systemic size gain between 3 to 4% linear size gain, which could be very small when you look, you can believe it's very small when you look at the curves, but when you do a bit of anthropomorphism, 3 to 4%, it's about 6 to 8 centimeters by just the intervention with the probiotics. So that is something that would be encouraging as a phenotype. So we confirmed also that the somatotropic axis was engaged by the bacteria, as we could see in monoassociated animals, this time conventional animals with the bacteria. We could see increased IGF-1 levels in the serum and increased production of IGF-1 by the liver of those animals. So again, we could see strain specificity effect because we repeated the protocol, this time by treating conventional animal with either placebo, the WGL strain, or the NISO 2877 strain. And as you can see, the growth rate of those animals is improved by the WGL strain, but not as improved as it is with WGL strain with the NISO strain. So again, we have a strain-specific strain in terms of weight gain, and also we could see a big difference in terms of size gain in this context in mice. So we were very happy to see that also in terms of IGF-1 level, the difference between the two strains was observed in the serum levels of IGF-1 in this context. So if you remember our fly work, clearly the cell wall of the bacteria seems to be really important in the Drosophila model to signal the host and to activate some signals to the host. So we wonder whether that was the same in this mammalian system. So what we did is to this time treat animals with either heat-killed or just the cell wall of the bacteria. So not gentry animals, but animals that have a microbiota. So a bit similar to this biochemical complementation I mentioned previously in flies. And what we could show is that the animals which were actually had a microbiota but were receiving the cell wall or the heat-inactivated bacteria WGL strain, they were putting on weight, but also size more than the strain that were either treated with a placebo or the cell wall or the heat-inactivated bacteria NISO strain. And that was also recapitulated with HF. So again, that was suggesting that really the cell wall is an important parameter in the plantarum positive effect on the host growth. And now capitalizing also again on the fly work, I mean, we could show in the fly that the peptidoglycan sensing machinery in the gut is involved. And here, what we did is just test genetic condition in the host where the sensing of the cell wall component in mammat is altered. So those are node or TLR receptors. And those guys are engaged by cell wall components in mammals. So we tested first in cultures, in cell cultures, whether the cell wall of plantarum WGL engage node 1, node 2, TLR2, or TLR4. And we could show that the cell wall of plantarum WGL engaged significantly node 2 signaling in human cell transfected with node 2, or TLR2, but not node 1 and TLR4. So then what we did is to generate an additional protocol where this time we were looking at mice which were knockout for either node 2 gene or MID88, which is the adapter of the TLR2, I mean, few TLRs, but including TLR2 in the context. So this is a paper that is currently in review, but we could show that treating MID88 knockout mice with Lactobacillus plantarum WGL upon undernutrition did not alter the beneficial impact of plantarum, so we could detect again weight gain and size gain in those mutant animals. Yet in the node 2 null mutant, we couldn't see anymore this beneficial effect. So this is I'm turning that in a short story, but we have additional work there going on, and we had a very nice referees that in the middle of the COVID pandemic were asking us to generate tissue-specific node 2 knockouts, of course, gut-specific, liver-specific, and macrophage-specific. So we are in the middle of that, and that's a pleasure for a flight geneticist as me to see how tricky it is to generate tissue-specific knockouts in mice, but that's a good challenge for a COVID period. Anyway, that little parenthesis closed. I'd like to wrap it up now and just provide the final take-home messages. So for this last part, especially, I mean, we've been developing now a preclinical model of low-protein diet-induced stunting. So we have mouse models where when you reduce protein content in the food, you have a stunting phenotype that is engaged, and we could show by impacting and treating in a daily manner those animals with isolates of Lactobacillus plantarum, we have somehow a buffer of the deleterious effect of the low nutrition. I'm not saying here that the bacteria is actually compensating for the loss. I mean, we are just rescuing few percent of the alteration. So we are really now thinking about combining probiotics with renutrition strategies, because that is actually the first thing you do when you're chronically undernourished. You refeed the animals or the kids. But we could show that we could show the proof of concept that the microbial intervention has an impact, and that impact at least partly goes through the somatotropic axis. We are really eager now to understand the basis of the strain specificity. And really, it's again all about cell wall sensing, it seems. And we could show in mammals that most likely the node 2 receptor is really engaged, and that maybe strain specificity comes from the cell wall sensing, either through node 2 or through other receptors. And we clearly have indications by looking at cell proliferation and some specific cell markers in the gut, in the small intestine, that those effects, like in flies, occurs in the small intestine and in enterocytes or other cell types in the small intestine of mouse models. Of course, there is an open avenue here to investigate the impact of those bacteria on host metabolism. We start to have phenotypes about insulin sensitivity, glucose control, glycemia control in those animals, which are exciting, but it's really the early days, so we are starting to investigate that. And as I previously mentioned, we are now developing preclinical models of chronic undernutrition followed by renutrition with either normal food or actually diabetic, like Western diet type of food, and combining that with probiotic intervention just to see what's the impact of renutrition and probiotic intervention for growth, but also for metabolism. So those are the avenues we are investigating now and developing in the lab with the team. And just a snapshot, and last minute, the snapshot of a paper we just published. We are slowly but surely moving from this one single strain approach, which was a very good model and has yielded some nice potential for that specific plantarum strain. But we've been publishing recently now a paper where we designed a mouse strain that was germ-free but reassociated with 15 members of the mouse microbiota, so purified culture of those mouse bacterial strain, mouse isolated bacterial strain. We reassociated those germ-free mice with this GM15, gnotobiotic mice 15 model, and we could show that this minimal microbial community recapitulates many of the metabolic and general physiological phenotypes of the animal. And for us in particular, we were interested by looking at growth. And we will be exploiting that model to dissect the ecology of this community and how the ecology of that microbial community influence the host physiology and vice versa, how the host environment influence the biology of that communities. And that is really probably the next few years of the lab, next many years of the lab. I will skip that and we'll just finish with the lab members and the key lab members over the last 10 years. Gilles Storelli and Beraer Kozar on the fly side, Renata Matos and Mariana Martino on the bacterial side, the recent student Theodore and postdoc Jessica on the holistic diet side, which were very prolific while they passed through the lab. A recent staff scientist that joined my lab on the mouse part and is expanding the whole mouse part of the lab, and a key collaborator, first lab member and now collaborator, Martin, that actually pushed us to go on the mouse side, and we keep collaborating with him. He has his lab now in Czech Republic. We had also the help of many important collaborators, Marie-Pierre Chapeau-Chartier for the cell wall analysis, Federica Calebro and David Da Silva for really the analysis of the genomes, bacterial genomes in the holistic diet paper, and Hubert Vidal for the physiology. I mean, he introduced us to the mouse physiology in this context. And the last story about the GM15 mice model has been developed with the BioAster Microbiology Technological Institute, which is next door to our institute in Lyon. And we've been developing gnotobiogy in Lyon thanks to them, while before gnotobiogy is done completely with Martin in Czech Republic. With that, I'll thank the sponsors that we had over the years, and I thank you for your attention, and I'm very happy to take any questions.
Thanks so much, François. That was almost an overdose of biology. Thank you very much for this amazing overview. And I personally, I appreciate that you started from the beginning, because it's a good sort of overview of what your contributions have been over the past few. I think there should be time for a few questions. And I will refrain from asking anything just in case. Okay, of course, Babis, please go ahead.
I thank you for the amazing talk. So I'm just wondering, so if you provide Drosophila with growth promoting bacteria, do they change essentially their feeding behavior?
A very good point. I should include those data. We carefully check their feeding behavior, and actually we couldn't detect any increase in food intake, ni transit time, or food. The food intake is not impacted. Yet we are now finding something very interesting, which I think would be part of the whole system, is that the gut of the associated larvae is about 20% longer than the gut of a germ-free larvae. Basically, this tells you that having the bacteria in the system, somehow, for a given size of larvae, when I speak that the gut is 20% larger, is longer for size match larvae, okay? So it means that there is an impact of the bacteria for the systemic growth of the animal, but also for tissue-specific growth. So we have a project going on in the lab currently where a lecturer actually in my lab with our student, she's actually trying to understand how the bacteria shape this gut growth phenotype, and we are dissecting the mechanism here. And we are testing whether the gut growth phenotype is actually causal or a consequence of the systemic phenotype. And the first results suggest that there is no direct link between the gut size and the systemic growth. So it's rather a relay, and we are actually now investigating that. And it seems that ecdysone is really important in this context, that you have an enhanced ecdysone production and signaling in a systemic manner when the bacteria is present, because you have more increased digestion. And that systemic ecdysone, for a reason that we are trying to understand now, has a specific impact on the gut to enhance gut growth. And that is actually amplifying the systemic effect, because you have a bigger gut, so you have a better absorption thanks to the bacteria. But that is not local effect, but it's really like a relay of the ecdysone. So, I mean, this is the working hypothesis. We have not yet demonstrated everything there, but we have some good, I believe, good loss of function studies of ecdysone transporters and ecdysone receptors in the gut that show that.
Mm-hmm. Have you guys ever done like experiment to mix two populations, like a fast growing or promoting strain together with a low, and look at evolution over generations instead of using only one?
No, we've not done that. We just did that for competition assays when we were doing experimental evolution to see whether one was outcompeting the other. I mean, whether the ancestral strain was outcompeted by the variant, because we could show that after few bacterial generation, the variant we isolated was actually taking the entire space of the niche. And when we did one-to-one colonization, of course, there was a fitness cost for the ancestral strain, and the evolved strain was really outcompeting that. So we did that for those specific contexts. We didn't do that in a general manner with growth promoting versus non-growth promoting. But what we did, well, we did it for looking at host phenotype, and the phenotype was always similar to the growth promoting strain phenotype. But we didn't check the relative proportion of each strain over time.
Okay, I'm intrigued with the oxidative stress, but I will ask you later when we talk.
Yes, but I mean, I can tell you that we don't understand it yet. We just
okay. We can talk later. Yeah.
Yeah. Sure.
All right. Any more questions? If not, I do have a few. So one of the things that I think strikes me more of what you've been telling us is this effect of not just Lactobacillus plantarum, but also just the biochemical complementation. So you can have just the cell wall, or I presume also with the polysaccharide in the first study, you can change the behavior of the fly, and I find it very fascinating, because you would, and also with the mice, right? Because I would expect that, naively perhaps, that if you have a bacteria in your gut that produce beneficial metabolites, the metabolites themselves would have the signaling properties, so that the host responds to the real thing, what is really interesting for. Whereas I presume that the nutritional value of the polysaccharide alone is close to nil. Certainly from the point of view of incorporating amino acids. So in a way, it's a bit like the fly is tricking, and the mouse is tricking itself into making a better profit of the existing diet.
So that's that. Yes, that makes sense. That's exactly it. I mean, my working, how I see the system is that you have basic nutritional and metabolic needs that are way above the threshold that the body is imposing, thanks to the endocrine regulation of the system. And the bacteria is somehow tricking the system by sending signal. It's not the bacteria that is tricking the system. It's the host is using microbial cues to let its endocrine system release the brakes on a condition that is suboptimal. Yet, and that's one layer of regulation, yet there are additional layers of regulation touching nutrition, because the bacteria also produce nutrients, and metabolic. And this is where we are not good at yet. We don't understand really much what's going on at the metabolic levels. And this is really somewhere we are going now, really the bacterial and the host metabolism, we really want to understand in the context of that. So you see, there are the 3 layers. The endocrine signals, basically it's the big breaks or the big go. Okay, insulin, IGF-1, ecdysone, all those big guys, hormones. Then you have the metabolism, the cross metabolism between the bacteria and the host. And that we don't understand yet. But I believe there is this layer. I mean, we have initial observations. And then there are the basic nutrient compensation and complementation that basically plantarum produce B vitamins, riboflavins, some essential amino acids that are important for the host. And when you have few of those in the diet, having a bit more thanks to the bacteria, it can only be helpful for the host. And that's the cumulative, the accumulation of the nutritional, the metabolic, and the endocrine signals that I believe make the bacterial effect so pronounced. So some are super important, but some are not sufficient. And that's why if you just provide the cell wall, maybe you release a bit of the break of the endocrine system, but you don't have the full picture. It's really pleiotropic. You don't have the nutrients. You don't have the anabolism kick, So you don't have systemic phenotype. Now, and I didn't illustrate that, if you take a germ-free animal, let's speak about flies, you provide cell wall on germ-free animals, growth is not affected. But when you look at digestive enzymes expression, for example, then those guys start to go up. Because the signal is there, but that is not sufficient to promote growth. Seems to be necessary, but not sufficient. So you see, and that's why, of course, it's the model I'm studying, so I love it. But I love this model because you have all the different layers and you can tackle them, see what is necessary. And so far, we found little mechanisms that are sufficient. It's really, you have to have different layers. One layer that we got is this amino acid, N-acetylated amino acid provision that actually on its own support growth promotion, which is quite of a phenotype. But you see, I really believe about this multi-layer nutrition, metabolism, and endocrine thing, which makes the system great to dissect the multi-layer system. And it's really what I call the molecular dialogue between the bacteria and the host, and between the host physiology. It's not just about nutrition. It's not just about metabolism.
Okay. So another question, now you've mentioned the N-acetyl amino acids. I presume that includes N-acetylcysteine.
Yes. And
where this is going, right?
Yeah. So the one that had a profound phenotype, it's N-acetyl glutamine. That was the one that was providing really a marked effect on growth. I'm not quite sure that N-acetylcysteine was N-acetylated cysteine was really decisive in terms of growth promotion, but it was part of the amino acids that are really enriched when we did a metabolism analysis of metabolomic analysis of the substrate, which were metabolites, which were produced by the strain. And we could really see many of those metabolites. And then we tested hat-back experiment with purified N-acetylated amino acid, and acetylated glutamine was really a driver of growth. So we suspected that was a sufficient one.
So you don't think that this protection against reactive oxygen species and oxidative stress could be mediated by acetylcysteine?
That's where, I don't want to put my fingers there, because, we had, this anabolism effect and this resistance to oxidative stress. It's purely hypothesis driven for now. It's purely like interpretation out of metabolomics data. And we could find some compounds which are annotated as antioxidant that were produced by the bacteria. I mean, we have not done much into showing that was really actually very important. We are digging into that. The only thing we've done is that in another paper, we could show that there is a low level of oxidative stress that is triggered by the bacteria because of the metabolism of the Lactobacillus, which have a lot of oxidative activity, an extracellular one actually. And that low level of oxidative activity in the gut is important for the host growth. That we've shown it by feeding NAC to the animal, like antioxidant to the animal. And actually that was freezing completely the beneficial effect of the bacteria. So we are trying to reconcile that. Others in the field have shown that there is also some NADPH oxidase which are engaged in the gut, which are producing growth burst in the gut, and that could be contributing also to some of the gut phenotypes. So we've not entirely revisited that, but it may be involved in the system.
So, okay, and one final question. So the Drosophila intestine is lined by chitin.
Yes.
So do you think that, so when you've mentioned this thing about the chitin from the larvae helping the bacterial culture, do you think the chitin might have function in keeping or at least maintaining the growth of the bacteria inside the gut?
Yeah, so in this context, our system is not ideal because our system, we use lab isolated strains which are not residing in the gut. So basically they are passing through, they are having their effect by releasing metabolites and several fragments, nutrients when they pass through. And we believe they are basically benefiting from the ketting monomers and the ketting debris in the fly food. Okay. Now, the lab of Will Lindington and the lab of Rich Teixeira in Lisbon in the US, they have isolated wild-derived lacto and aceto strains, which are actually now residing in the gut, persisting in the gut, especially in the anterior part, in the foregut. And they showed that those bugs, they are attached to the ketting there. So I very much believe that what we could find in the fly food is also occurring in the gut, in the foregut, where basically they form biofilm and they probably feed on those ketting monomers.
Fantastic. Okay, I think I'm going to stop now, otherwise we will pass the time now. And I would like to thank you again for this fantastic talk.
Thank you. Thanks very much.
Someone in the chat who has someone who was just saying thanks.
Thank you.
All right. So, well, I'll see you all guys next week at the same time. And
okay, we are going to continue now.
Patrick,
yeah.
Shall I go to the office?
Okay, to my office.
Or you can come to mine. That's okay, fine.
I'll bring my talk is big. I can't.
I'll go to your office.
Okay.
My slides in box.
Okay, thanks.
So we disconnect from this one.
And we can just stay on.
We will disconnect from this session. I think it's better because then,
because
you know, some people. This is the thing. Some people thought. Sorry, François. This is, but the people who. Some people think that this talk was at 2 because one of the talks at 1 has been changed, and I fear that someone may pop in.
Okay, don't worry. So we go to the next link that you sent us this morning.
Okay.
Yes, please. Yes.
Okay.
So I'll see you in a minute.
Give me 2 minutes, and I'll be back in 2 minutes.
Of course,
okay?
of course. Yeah.
See you soon.
See you soon.